So this chapter lends itself to be broken up into bits and pieces, so this one's gonna be the effects of outer influences, environmental influences, if you will. We're gonna look at the effects of temperature on enzyme activity. We're gonna look at the effects of pH on enzyme activity. And I may ask about both, I may ask about one. If I ask about one, don't answer about the other one. That's a very common mistake that people make on exams. I ask a pH question, they give me a temperature answer. I ask a temperature question, they give me a pH answer. Don't do that. I'm not going to put a term on the reaction rate. I'm going to label the axis that way. Again, no actual measurements because the different enzymes will react to different temperatures. Is that enzymes that are in us are going to have a different temperature response curve than enzymes that are in ice worms that live on the surface of glaciers. And the curve, I'm giving you an idealized curve and then talk a little bit about variations. But this is very much an idealized curve that we're going to look at. Your odds of getting a nice bell, I mean, a bell shape you'll get, but whether it's going to be a nice balanced bell is probably pretty low. Let me get my little pointer. So you start out where everything's too cool. Remember, the hotter it is, the more the particles move. Well, the cooler it is, the slower the particles move. And at a certain point, they're not moving fast enough to really cause the reaction rate to go, right? Q10 and all that. Chances of it being actually zero, or again, it's an idealized curve. But as the temperature gets warmer, the particles start to hit each other, which means the substrates start to hit the enzymes start to cause the reaction to speed up, and the products are produced. And how do you measure the reaction rate? How many products are we getting? So the temperature goes up. You hit a point where, and this is always used using Vmax. You've hit your Vmax. What happens as it gets warmer? Remember, particles are not just moving in response to, something just made a noise. Not just moving in response to temperature. The large molecules, like the enzymes, are also shaking. And what's giving the enzymes their shape a lot of times are hydrogen bonds inside, and you can pop those with enough shaking. So as it gets warmer, you don't have as many enzymes working because some of them have changed into a shape where they're not functional. You've changed the shape of the active site, they're not grabbing stuff anymore. And the warmer it gets, the more enzymes are denatured, remember denatured? And so they're not working, so the reaction rate falls. You need the enzymes in order to get the reaction rate rolling. Fewer and fewer enzymes mean the reaction rate falls. And eventually, it's so hot that the enzymes are all of your enzymes are not shaped right. And they're not gonna reform either because it's too doggone hot. And again, this rate probably would top out here at some level where the reaction would be driven purely by the heat, but idealized curve. There's a point on the temperature gauge where this is running as well as it can run. That is the optimal or the optimum temperature. Both terms are used, both terms apply. Gee, you want to take a guess at what the optimal temperature is for most of the enzymes in us? Yeah, it's in the somewhere around 97, 98 degrees Fahrenheit, somewhere around 37, 38 degrees Celsius. Is it over time we have, as we've generated internal heat, we have selected through enzymes that are more effective working at that set temperature. I mean, sometimes weird stuff happens. Is that sperm production, apparently in our ancestors, the testes right underneath the surface. And rather than select enzymes that worked well at the temperature we were all settling on, those enzymes worked really well at a slightly lower temperature. And so, you know, with evolution, you don't get what makes sense, you get what you get. In the case of mammals, the site of sperm production was actually externalized, where those temperatures could be kept cooler than the internal body temperature, is that they were moved out into a sac, basically, where the optimal temperature for sperm production enzymes is somewhere down around 93, 94 degrees Fahrenheit. And in testes, it can be kept that way. In mammals that don't have hanging testes, they have this complex system of blood circulation where they pull cooled blood from the surface down to circulate around the testes. Because if the testes come up to normal body temperature, the enzymes don't work as well. You get up into this range, and sperm production is compromised. But our curve would be a very tight curve. It would be really narrow because a couple of degrees too cool, the enzymes don't work real well at all. A couple of degrees too warm, you run too high a temperature, your enzymes don't work too well. These curves also are different for different enzymes. Enzymes that have to work out here have got to work at a lower temperature than your core body temperature. So their curves would be broader. And in some cases you get that might be a curve for a local plant that needs to be able to do whatever this is doing in the spring when it gets warm enough, in the middle of the summer when it's really hot, in the fall when it's cooling off. May not work that well in the wintertime, but for the broad range of what it needs that enzyme to be working. It may not need it to be working super, super well. It may not have a really high optimal rate, but as long as it can work properly. What you find in some animals, here, this is a better curve. This was a little tricky. These are two enzymes doing the same job. One of them has an optimal temperature that fits burrow temperature. The other one has an optimal temperature that hits midday temperatures. So during the warmth of the midday, this enzyme is active. During the night when they're maybe hunting in burrows, or they're active at night, or they're just doing something, or they're just in their burrow, then this enzyme is active. And there's often a little trough in between where this animal's like, say, a snake needs to go out and sun itself. It wants to get through this trough of low activity as quickly as possible and get its body temperature up to here so the enzyme is working better. So enzymes work a whole bunch of different ways. A lot of times these curves are lopsided, where with us, how it drops off on each side of the cool side and the warm side may not be as precipitous on one side as on the other side. I would think it drops off probably at the high end quicker than at the low end, even though hypothermia is a bad thing. Well, I'm going to leave that there. Oh. So the rate drops off on both sides of the optimal. I didn't mention this. As the enzymes cool off, remember this shaking? as you warm them up. Well, as you cool them, they compress somewhat, right? Is that the particles moving inside the protein are not shaking as much, and so the bonds are going to pull them a little bit closer together. That means that the active site is going to get a little tighter. The affinity is going to drop a bit. And that's another reason why this drops off with cooler temperatures. It isn't just that the particles are moving slowly, it's that the enzymes can't grab and release quite as quickly, not just because the particles aren't there to grab and release as much, but also it's a little bit tighter to fit them in, it's a little bit harder to let go of them. So there's two reasons why this drops off on the cold side. And kind of one reason, the activity of the particles, why it drops off on the high side. But you lose the nature of the enzyme in both directions. Okay, back to this. Remember I said don't confuse the temperatures with the pH? This is the pH. With the rate going up there. And of course, this also has an optimal or an optimum. Again, idealized curve. Typically, these curves are much narrower. Is that there's an optimal pH, and if you move it in either direction for biological systems, tends to drop off pretty doggone fast. Now, the question is, why are the enzymes not working well on either side of this particular point? Well, as the pH goes up, what do you get more of? pH is the relationship between, well, it's measurement of hydrogen ions, but it's the relationship between hydrogen ion and hydroxide ion concentrations. And those are very small charged particles. They can get inside a protein and disrupt the hydrogen bonds that keeps the protein in the right shape. So there's going to be a concentration of hydrogen and hydroxides where that protein is going to be at its best possible shape all the time. You throw a few more hydroxides at it, the more and more you do, the more and more it loses shape. Throw some hydrogens at it, the more you do, the more it loses shape. Now that pH is not always going to be neutral, right? As a lot of our body tissues have a slightly, I was going to say alkaline, slightly basic pH. That tells you how old I am. And so our optimal pH for a lot of stuff, especially blood stuff, tends to be a little bit above neutral. That one enzyme in our stomachs, that as the stomach acid disrupts the proteins, clips it, and the optimal pH is, that's my wife calling, so I know I can ignore it. And I'm almost done with this thing, so I'll let it ring. Sorry if it's distracting you. That particular enzyme works really well, way down at a pH of about a 3, is that the hydrogen bonds stabilize it rather than disrupt it. And, sorry, distracting that it stopped. So this is not always going to be a neutral pH. For some environments that run a little acidic, the things that live there tend to have an optimal pH on the acidic side. Things that run more basic, their optimal pH is on the basic side. Keep in mind, in neutral systems, with the exception of stomach acid, is that doesn't tend to be a huge range of good pHs where things work well. So that's why I said these curves are usually a lot tighter than this idealized bell that we've got. Okay, that's a good place to stop. Let me just take a look, make sure that I've covered everything, and we'll stop that for temperature and pH effects.